Neural oscillation is rhythmic, a repetitive neural activity in the central nervous system. Neural tissue can generate oscillatory activity in many ways, driven either by mechanisms within individual neurons or by interactions between neurons. In individual neurons, oscillations can appear either as oscillations in membrane potential or as rhythmic patterns of action potentials, which then produce oscillatory activation of post-synaptic neurons. At the level of neural ensembles, synchronized activity of large numbers of neurons can give rise to macroscopic oscillations, which can be observed in an electroencephalogram. Oscillatory activity in groups of neurons generally arises from feedback connections between the neurons that result in the synchronization of their firing patterns. The interaction between neurons can give rise to oscillations at a different frequency than the firing frequency of individual neurons. A well-known example of macroscopic neural oscillations is alpha activity. Neural oscillations were observed by researchers as early as 1924. More than 50 years later, intrinsic oscillatory behavior was encountered in vertebrate neurons, but its functional role is still not fully understood. The possible roles of neural oscillations include feature binding, information transfer mechanisms, and the generation of rhythmic motor output. Over the last decades, more insight has been gained, especially with advances in brain imaging. A major area of research in neuroscience involves determining how oscillations are generated and what the roles are. Oscillatory activity in the brain is widely observed at different levels of observation and is thought to play a key role in processing neural information. Numerous experimental studies support a functional role of neural oscillations. A unified interpretation, however, is still lacking. Overview: neural oscillations are observed throughout the central nervous system at all levels, and include spike trains, local field potentials, and large-scale oscillations which can be measured by electroencephalography. In general, oscillations can be characterized by the frequency, amplitude, and phase. These signal properties can be extracted from neural recordings using time-frequency analysis. In large-scale oscillations, amplitude changes are considered to result from changes in synchronization within a neural ensemble, also referred to as local synchronization. In addition to local synchronization, oscillatory activity of distant neural structures can synchronize. Neural oscillations and synchronization have been linked to many cognitive functions such as information transfer, perception, motor control, and memory. Neural oscillations have been most widely studied in neural activity generated by large groups of neurons. Large-scale activity can be measured by techniques such as EEG. In general, EEG signals have a broad spectral content similar to pink noise, but also reveal oscillatory activity in specific frequency bands. The first discovered and best-known frequency band is alpha activity that can be detected from the occipital lobe during relaxed wakefulness and which increases when the eyes are closed. Other frequency bands are delta, theta, beta, and gamma frequency band, where faster rhythms such as gamma activity have been linked to cognitive processing. Indeed, EEG signals change dramatically during sleep and show a transition from faster frequencies to increasingly slower frequencies such as alpha waves. In fact, different sleep stages are commonly characterized by the spectral content. Consequently, neural oscillations have been linked to cognitive states, such as awareness and consciousness. Although neural oscillations in human brain activity are mostly investigated using EEG recordings, they are also observed using more invasive recording techniques such as single-unit recordings. Neurons can generate rhythmic patterns of action potentials of spikes. Some types of neurons have the the tendency to fire at particular frequencies, so-called resonators. Bursting is another form of rhythmic spiking. Spiking patterns are considered fundamental for information coding in the brain. Oscillatory activity can also be observed in the form of subthreshold membrane potential oscillations. If numerous neurons spike in synchrony, they can give rise to oscillations in local field potentials. Quantitative models can estimate the strength of neural oscillations in recorded data. Neural oscillations are commonly studied from a mathematical framework and belong to the field of neurodynamics, an area of research in the cognitive sciences that places a strong focus upon the dynamic character of neural activity in describing brain function. It considers the brain a dynamical system and uses differential equations to describe how neural activity evolves over time. In particular, it aims to relate dynamic patterns of brain activity to cognitive functions such as perception and memory. In very abstract form, neural oscillations can be analyzed analytically. When studied in a more physiologically realistic setting, oscillatory activity is generally studied using computer simulations of a computational model. The functions of neural oscillations are wide-ranging and vary for different types of oscillatory activity. Examples of the generation of rhythmic activity such as a heartbeat and the neural binding of sensory features in perception, such as the shape and color of an object. Neural oscillations also play an important role in many neurological disorders, such as excessive synchronization during seizure activity in epilepsy or tremor in patients with Parkinson's disease. Oscillatory activity can also be used to control external devices in brain-computer interfaces, in which subjects can control an external device by changing the amplitude of particular brain rhythmics. Physiology. Oscillatory activity is observed throughout the central nervous system at all levels of organization. Three different levels have been widely recognized: the microscale, the mesoscale, and the macroscale. Microscopic neurons generate action potentials resulting from changes in the electric membrane potential. Neurons can generate multiple action potentials in sequence, forming so-called spike trains. These spike trains are the basis for neural coding and information transfer in the brain. Spike trains can form all kinds of patterns, such as rhythmic spiking and bursting, and often display oscillatory activity. Oscillatory activity in single neurons can also be observed in subthreshold fluctuations in membrane potential. These rhythmic changes in membrane potential do not reach the critical threshold and therefore do not result in an action potential. They can result from post-synaptic potentials, from synchronous inputs, or from intrinsic properties of neurons. Neuronal spiking can be classified by their activity patterns. The excitability of neurons can be subdivided in Class 1 and 2. Class 1 neurons can generate action potentials with arbitrarily low frequency depending on the input strength, whereas Class 2 neurons generate action potentials in a certain frequency band, which is relatively insensitive to changes in input strength. Class 2 neurons are also more prone to display subthreshold oscillations in membrane potential. Mesoscopic E group of neurons can also generate oscillatory activity. Through synaptic interactions, the firing patterns of different neurons may become synchronized and the rhythmic changes in electric potential caused by their action potentials will add up. That is, synchronized firing patterns result in synchronized input into other cortical areas, which gives rise to large amplitude oscillations of the local field potential. These large-scale oscillations can also be measured outside the scalp using electroencephalography and magnetoencephalography. The electric potentials generated by single neurons are far too small to be picked up outside the scalp. scale, and e.g. omega activity always reflects the summation of the synchronous activity of thousands or millions of neurons that have similar spatial orientation. Neurons in a neural ensemble rarely all fire at exactly the same moment, i.e. fully synchronized. Instead, the probability of firing is rhythmically modulated such that neurons are more likely to fire at the same time, which gives rise to oscillations in the mean activity. As such, the frequency of large-scale oscillations does not need to match the firing pattern of individual neurons. Isolated cortical neurons fire regularly under certain conditions, but in the intact brain, cortical cells are bombarded by highly fluctuating synaptic inputs and typically fire seemingly at random. However, if the probability of a large group of neurons is rhythmically modulated at a common frequency, they will generate oscillations in the mean field. Neural ensembles can generate oscillatory activity endogenously through local interactions between excitatory and inhibitory neurons. In particular, inhibitory interneurons play an important role in producing neural ensemble synchrony by generating a narrow window for effective excitation and rhythmically modulating the firing rate of excitatory neurons. Macroscopic neural oscillation can also arise from interactions between different brain areas. Time delays play an important role here. Because all brain areas are bidirectionally coupled, these connections between brain areas form feedback loops. Positive feedback loops tends to cause oscillatory activity, which frequency is inversely related to the delay time. An example of such a feedback loop is the connections between the thalamus and cortex. This thalamocortical network is able to generate oscillatory activity known as recurrent thalamocortical resonance. The thalamocortical network plays an important role in the generation of alpha activity. Mechanisms. Neuronal properties. Scientists have identified some intrinsic neuronal properties that play an important role in generating membrane potential oscillations. In particular, voltage-gated ion channels are critical in the generation of action potentials. The dynamics of these ion channels have been captured in the well-established Hodgkin-Huxley model that describes how action potentials are initiated and propagated by means of a set of differential equations. Using bifurcation analysis, different oscillatory varieties of these neuronal models can be determined, allowing for the classification of types of neuronal responses. The oscillatory dynamics of neuronal spiking as identified in the Hodgkin-Huxley model closely agree with empirical findings. In addition to periodic spiking, sub-threshold membrane potential oscillations, i.e. resonance behavior that does not result in action potentials, may also contribute to oscillatory activity by facilitating synchronous activity of neighboring neurons. Like pacemaker neurons in central pattern generators, subtypes of cortical cells fire bursts of spikes rhythmically at preferred frequencies. Bursting neurons have the potential to serve as pacemakers for synchronous network oscillations, and bursts of spikes may underlie or enhance neuronal resonance. Network properties. Apart from intrinsic properties of neurons, network properties are also an important source of oscillatory activity. Neurons communicate with one another via synapses and affect the timing of spike trains in the post-synaptic neurons, depending on the properties of the connection, such as the coupling strength, time delay, and whether coupling is excitatory or inhibitory. The spike trains of the interacting neurons may become synchronized. Neurons are locally connected, forming small clusters that are called neural ensembles. Circuits certain network structures promote oscillatory activity at specific frequencies. For example, neuronal activity generated by two populations of interconnected inhibitory and excitatory cells can show spontaneous oscillations that are described by the Wilson-Cowan model. If a group of neurons engages in synchronized oscillatory activity, the neural ensemble can be mathematically represented as a single oscillator. Different neural ensembles are coupled through long-range connections and form a network of weakly coupled oscillators at the next spatial scale. Weakly coupled oscillators can generate a range of dynamics including oscillatory activity. Long-range connections between different brain structures, such as the thalamus and the cortex, involve time delays due to the finite conduction velocity of axons. Because most connections are reciprocal, they form feedback loops that support oscillatory activity. Oscillations recorded from multiple cortical areas can become synchronized to form large-scale brain networks, whose dynamics and functional connectivity can be studied by means of spectral analysis and Granger causality measures. Coherent activity of large-scale brain activity may form dynamic links between brain areas required for the integration of distributed information. Neuromodulation, in addition to fast direct synaptic interactions between neurons forming a network, oscillatory activity is modulated by neurotransmitters on a much slower time scale. That is, the concentration levels of certain neurotransmitters are known to regulate the amount of oscillatory activity. For instance, GABA concentration has been shown to be positively correlated with frequency of oscillations in induced stimuli. A number of nuclei in the brain stem have diffuse projections throughout the brain influencing concentration levels of neurotransmitters such as norepinephrine, acetylcholine and serotonin. These neurotransmitter systems affect the physiological state, e.g., wakefulness or arousal, and have a pronounced effect on amplitude of different brain waves, such as alpha activity. Mathematical description. Oscillations can often be described and analyzed using mathematics. Mathematicians have identified several dynamical mechanisms that generate rhythmicity. Among the most important are harmonic oscillators, limit cycle oscillators, and delayed feedback oscillators. Harmonic oscillations appear very frequently in nature. Examples are sound waves, the motion of a pendulum, and vibrations of every sort. They generally arise when a physical system is perturbed by a small degree from a minimum energy state, and are well understood mathematically. Noise-driven harmonic oscillators realistically simulate alpha rhythm in the waking EEG as well as slow waves and spindles in the sleep EEG. Successful EEG analysis algorithms were based on such models. Several other EEG components are better described by limit cycle or delayed feedback oscillations. Limit cycle oscillations arise from physical systems that show large deviations from equilibrium, whereas delayed feedback oscillations arise when components of a system affect each other after significant time delays. Limit cycle oscillations can be complex, but there are powerful mathematical tools for analyzing them. The mathematics of delayed feedback oscillations is primitive in comparison. Linear oscillators and limit cycle oscillators qualitatively differ in terms of how they respond to fluctuations in input. In a linear oscillator, the frequency is more or less constant, but the amplitude can vary greatly. In a limit cycle oscillator, the amplitude tends to be more or less constant, but the frequency can vary greatly. A heartbeat is an example of a limit cycle oscillation in that the frequency of beats varies widely, while each individual beat continues to pump about the same amount of blood. Computational models adopt a variety of abstractions in order to describe complex oscillatory dynamics observed in brain activity. Many models are used in the field, each defined at a different level of abstraction and trying to model different aspects of neural systems. They range from models of the short-term behavior of individual neurons, through models of how the dynamics of neural circuitry arise from interactions between individual neurons, to models of how behavior can arise from abstract neural modules that represent complete subsystems. Single neuron model. A model of a biological neuron is a mathematical description of the properties of nerve cells, or neurons, that is designed to accurately describe and predict its biological processes. The most successful and widely used model of neurons, the Hodgkin-Huxley model, is based on data from the squid giant axon. It is a set of nonlinear ordinary differential equations that approximates the electrical characteristics of a neuron, in particular the generation and propagation of action potentials. The model is very accurate and detailed, and Hodgkin and Huxley received the 1963 Nobel Prize in Physiology or Medicine for this work. The mathematics of the Hodgkin-Huxley model are quite complicated, and several simplifications have been proposed, such as the Fitzhugh-Nagumo model and the Hindmarsh-Rose model. Such models only capture the basic neuronal dynamics, such as rhythmic spiking and bursting, but are more computationally efficient. This allows the simulation of a large number of interconnected neurons that form a neural network. Spiking model. A neural network model describes a population of physically interconnected neurons or a group of disparate neurons whose inputs or signaling targets define a recognizable circuit. These models aim to describe how the dynamics of neural circuitry arise from interactions between individual neurons. Local interactions between neurons can result in the synchronization of spiking activity and form the basis of oscillatory activity. In particular, models of interacting pyramidal cells and inhibitory interneurons have been shown to generate brain rhythms such as gamma activity. Neural mass model. Neural field models are another important tool in studying neural oscillations and are a mathematical framework describing evolution of variables such as mean firing rate in space and time. In modeling the activity of large numbers of neurons, the central idea is to take the density of neurons to the continuum limit, resulting in spatially continuous neural networks. Instead of modeling individual neurons, this approach approximates a group of neurons by its average properties and interactions. It is based on the mean field approach, an area of statistical physics that deals with large-scale systems. Models based on these principles have been used to provide mathematical descriptions of neural oscillations and EEG rhythms. They have, for instance, been used to investigate visual hallucinations. Keira-Moto model. The Keira-Moto model of coupled phase oscillators is one of the most abstract and fundamental models used to investigate neural oscillations and synchronization. It captures the activity of a local system by its circular phase alone, and hence ignores the amplitude of oscillations. Interactions amongst these oscillators are introduced by a simple algebraic form and collectively generate a dynamical pattern at the global scale. The Keira-Moto model is widely used to study oscillatory brain activity, and several extensions have been proposed that increase its neurobiological plausibility. For instance, by incorporating topological properties of local cortical connectivity. In particular, it describes how the activity of a group of interactioning neurons can become synchronized and generate large-scale oscillations. Simulations using the Keira-Moto model with realistic long-range cortical connectivity and time-delayed interactions reveal the emergence of slow, patterned fluctuations that reproduce resting-state bold functional maps, which can be measured using fMRI.